In this multiple choice question, from the given option, they are directing us to specify the wrong option, illustrating chromosomal DNA analyzing using southern hybridization technique. For this, let me understand the sequential step in which way it is executed in southern hybridization technique. After labeling the DNA probe and the suspect DNA, we are introducing the radio labeled DNA into the gel electrophoresis. Once the DNA movement is completed, we are executing the blotting process to execute the double stranded DNA formation so that we can confirm the concerned suspect is culprit or not. That means we are doing the autoradiographic technique by exposing the double stranded DNA to the X-ray film. The exposed double stranded DNA over the X-ray film, if it is creating the spot, we can understand that is a radio labeled double stranded DNA. Like this we can confirm, so that we can understand the concerned parent of the respective child in the child dispute case. Like this, we are executing southern hybridization technique in most of the significant evaluation technique, but we are not using PCR in southern hybridization technique. So we are choosing option D as a correct answer to secure 4 mark. What are the points we told with this picture that we are giving as a sentence wise narration to have a recap and also to take the NEET notes for future reference. PCR is a method of enzymatic DNA replication that does not require the use of living creature like yeast or E. coli. It is frequently employed in biological and medical research laboratory for a range of purposes, such as genetic fingerprinting identification and the detection of the inherited disorder. With this specification, we choose an option D as a correct answer to secure 4 mark.